Hello, and welcome back to this tutorial for Astrolabe's Cell Subset Navigator. In this video, I'm going to show the MDS Map visualization. What you see here is our multidimensional scaling, or MDS Map. Each bubble in this map represents a cell subset identified by Astrolabe using the markers I defined in my panel. The size of each bubble corresponds to the median frequency of that subset across all of my experiments samples. There are a few ways I can navigate this data. I can hover over the bubbles to see the cell subset name and click to select, or I can use this menu bar to select the subsets I am interested in. Another thing I can do to add clarity to this map is color code it according to certain markers. So if I color code by CD3, I can see this cluster of T cells are shaded in a darker green. This data is hierarchical. If I want to examine T cells specifically, I could select them individually, or I could select Compartment, which is going to give me a more high-level view of my data. When I select T cells here, all of my T cells will also be selected in the Assignment tab. Likewise, if I want a more granular view of my data, I can select the profiling level here. This visualization shows me the subsets Astrolabe has identified via its unsupervised clustering algorithm. So for example, if I want to examine my CD4 positive, CD8 positive T cells, I can select them here, go to the profiling level, I have to scroll over a little, and see Astrolabe has found subsets of these cells with high or low expression of CCR4, CCR7, and PD1. These views are all interconnected, so if I selected more subsets here, I will see the container subsets also selected back at the assignment level. Next in this series, I will show how we can further explore these cell subsets.